Radioactive transduction. Radioactivity is a result of nuclear reaction. As a result of nuclear reaction, there is a spontaneous emission of radiation. This radiation is of two types. One is in the form of particles and other is in the form of high energy photons. That it means that are particles that are released due to the atomic reaction, due to the nuclear reaction, are called radioactivity. The radioactive decay is a random process, and this process occurs at an individual atom level. Now, what types of radiation particles are there? Number one is alpha particle. They carry a positive charge. Alpha particle is made up of two protons and two neutrons. They are bound together. Beta particles carry a negative charge, and beta particles are high energy electrons. In case of gamma, gamma rays are neutral. Gamma rays are waves of electromagnetic energy or photons. Now, the first time the radioactive transduction principle was used in radioimmunoassay. Radioimmunoassays are first used for the detection or for the estimation of hormone insulin. In this principle, a radioactive antigen competes with a non-radioactive antigen for a fixed number of antibody or receptor binding site. In this case, we know that for there was an immobilization process. For the immobilization, polyethylene glycol was used, in which those samples which bound with the antibody and antigen did not immobilize on the surface of polyethylene glycol, and those that the leftover antigens bound to the polyethylene glycol. And this was the main principle. In this case, we used the radio labeled material. The material that was used was iodine 125. It was mixed with the radioactive with the antigen, and it is assumed that the tyrosine, or mainly the tyrosine amino acid of the antigen protein, binds with the iodine, and that is how the antigen become radioactive. And this radioactive antigen was reacted with the antigen that is not treated with radioactivity, and then there is a competition between the two, and that is how the radioimmunoassay works. In the radioimmunoassay, first with the low concentration, we have very high radioactivity, and as we increase the concentration of our antigen that is non-radioactive, the concentration of the radioactive counts decays, as shown in this graph. So this is the basic principle for the radioactivity, and the immobilization polyethylene glycol is very important in this radioactive transduction. The radioactive transduction was also successfully used in molecular biology procedures, such as in case of DNA sequencing. In case of DNA sequencing, sulfur S35 was successfully used to detect the labeled nuclear for the DNA sequencing. And in autoradiograph radioactivity, sometimes P32, and it means phosphorus radioactive phosphorus was used for labeling the DNA and protein, and a number and these techniques were successfully used for many years. But now, due to the availability of different safe techniques, these radioactive techniques are no more in use then. The first thing is the safety level. Several disadvantages are associated, particularly regarding the technical and safety issues, and this may be the main reason that the radioactive transduction principles are not used in the biosensor technology.